So we're going to return to the boreal forest for a little while, and I'm going to tell you about northern peatlands, which are the world's largest terrestrial carbon store. They actively accumulate carbon because they have low decomposition rates. These decomposition rates are maintained through a high water table, obviously low temperatures, and poor nutrient conditions. And for the most part, this system is a feedback stable state. So the above ground communities are dominated by sphagnum mosses, and the sphagnum maintain this high water table. They create acidic conditions, they keep soil temperatures low, and they basically suppress decomposition in a way that allows for carbon to accumulate. Now below ground in the peat soil environment, we have relatively slow-growing, long-lived communities, microarthropods, mainly mites, as well as fungi that are relatively adapted to this low pH system and feed sort of very slowly on some of this recalcitrant carbon. However, there are some apparent changes for northern peatlands. Changes in atmospheric CO2, changes in temperature, changes in moisture regimes. And these are all expected to affect directly both the above ground as well as the below ground communities in a way that's going to directly alter decomposition rates. So specifically, elevated atmospheric CO2, elevated temperature are expected to enhance vascular plant growth. This is expected to then release that competitive stronghold that the sphagnum mosses have. And this is going to have cascading effects into the below ground system through changes in litter quality and litter quantity, as well as labile carbon through root exudates. So below ground, we would actually expect that these extra carbon sources, in addition to direct effects of elevated temperature, and more habitable pore space due to a lower water table is going to create favorable conditions for the below ground biodiversity for the most part. And the below ground communities are the ones that are going to directly drive decomposition rates. And this is going to have feedback effects back up to the above ground communities through changes in water quality and nutrient availability. So my research team decided to address the effects of elevated temperature, elevated CO2, and lower water table on a sphagnum-dominated boreal peatland using a large-scale mesocosm experiment. So we collected a metric ton of peat from a boreal forested poor fen peatland in central Canada. We collected it as 100 intact vegetated monoliths. And we brought this back to our research institution in southern Ontario, and we exposed it to future climate scenarios over 18 months in a climate change facility that we call the Biotron. So the Biotron is a building, even though it sounds like it should be a robot. On the top of the Biotron, we have 6 experimental greenhouses that can simulate a whole range of future climate change scenarios. The climate change scenarios that we selected were based on the IPCC predictions for the boreal forest over the next 50 to 100 years. So specifically, these were elevated temperatures between +4 and +8 degrees Celsius above current ambient conditions, a near doubling in atmospheric CO2, and a more sporadic but lower precipitation, resulting in a lower water table by about 20 centimeters. So our 100 mesocosms were allowed to recover for about 4 months prior to going into experimental treatment. During this time, we fit them with a drainage system at the bottom, which also allowed us to maintain our water tables later in the experiment. And at the start of the experiment, after 4 months, we destructively sampled 16 of these mesocosms. And the purpose, well, my interest was in mostly the above ground and the below ground biodiversity, but also a few ecosystem level processes we were interested in as well, mainly relating to carbon. So I'll talk about decomposition today, and I'll also talk about dissolved organic carbon quantity and quality in pore waters as well. So these mesocosms went into their experimental treatment. We had 7 replicates for each of our combination of treatments. And each of these mesocosms, we had 84 that went into treatment. They were fitted with 3 litter bags that allowed us to destructively sample the litter bags, but not the meat the peat mesocosms at 4 months, 8 months, and 12 months. And then we fully destructively sampled at 18 months. And so our first prediction was about this above-ground biodiversity and changes in the above-ground vegetation. Specifically, we thought that elevated CO2 and elevated temperature was going to enhance vascular plant growth at the expense of the sphagnum cover. And for the most part, our predictions held. So we found under elevated temperature, in particular, declines in sphagnum moss over the first 12 months. On the left-hand side here, and this coincided with an increase in vascular plants, in particular a small graminoid species called Carex disperma. The effects of CO2 in this case were actually quite weak. We only saw the effect of CO2 on one of our plant species, which was another Carex magellanicum. But the results were apparent and dramatic. So visually, the mesocosm up at the top, this is mesocosm number 36 before the experimental treatments. Below is the same mesocosm after 4 months, and after 8 months, and after 12 months. So these changes were rather dramatic, at least for elevated temperatures at +8, and we expected then that this was going to have some effects on the below ground biodiversity as well. So our predictions for the below ground biodiversity, particularly the microfauna, or sorry, the mesofauna and the fungi, is that elevated temperature and lower water table was going to create more habitable pore space. It was going to create favorable conditions. It was going to release some of the metabolic and reproductive constraints. And so it was actually going to enhance the abundance of some of the microarthropods. At the same time, I also thought that even though it's quite cool in the boreal forest, elevated temperatures in the summer do reach near lethal temperatures. So I did think that elevated temperatures were going to impact the richness. So our results in this case somewhat held against our predictions. So on the left-hand side, we have average abundance of microarthropods, and on the left-hand side of the green bar is the average abundance, so number of individuals per gram dry weight of microarthropods pre-treatment. And you can see this dramatic increase after 18 months of elevated temperature, where the average abundance of microarthropods increased, basically nearly doubled with each of our temperature treatments. And this did coincide with enhanced reproductive rates. The proportion of juveniles also pretty much doubled in each of our temperature treatments. The story for richness was a little bit different. We didn't actually see any of the effects of temperature. In this case, there was very few richness effects. The only decrease in richness that we did see was under a permanently saturated water table. However, when we look at who is there in the community, so community composition, this was completely driven by temperature. And so this is an NMDS ordination plot. The data points represent a community in a particular mesocosm. Data points that are close together represent that these communities are more similar to each other than data points that are further away. And you can see the +8 experimental treatments, the communities are becoming much more similar to one another. So they're becoming homogenized, as well as moving away from the communities at +4 in ambient conditions. And we know that this isn't due to non-random loss of any particular group. In fact, it's due to an increase in the relative abundance of particular groups. And these groups for the orobatid mites were small-bodied, asexually reproducing, in the families Sectobelbidae and Brachychthonidae. So those are the patterns that are happening in the microarthropods. We find very similar trends happening in the fungi as well. So this is some preliminary data. This is fungi sampled from litter bags after just 4 months. And similar patterns here. Temperature is the main driver of fungal community composition. And again, at that +8 degree treatment, we find this homogenizing effect. And my colleague Asma Manahed, who's performing the fungal data, tells me again, it's not due to any particular loss of organisms, it's actually due to a relative abundance, a relative increase in the abundance of saprotrophic fungi. So that's the biodiversity aspect of this. We also looked at ecosystem function, and we particularly thought that what was happening below ground, as well as a little bit what was happening above ground, was going to affect ecosystem level processes, particularly decomposition rates. And so we thought decomposition was going to be driven mainly by temperature, but also a little bit by moisture, water table as well. And so pretty straightforward, our decomposition results did follow along with our predictions. Both temperature and lowered water table enhanced decomposition. It turns out that lower water table was actually a bigger driver than temperature, but you can see in both the saturated conditions as well as the lower water table conditions, decomposition rates increased with elevated temperature, and in particular in that +8 degree treatment as well. When we look at the actual carbon in the system, so this is dissolved organic carbon coming out in pore waters that we collected as leachate. Again, we see when we look at the total amount of carbon coming out of the system, it's again related to that temperature treatment, and in particular the +8 degrees above ambient. And so the thing about the DOC quantity is that this involves obviously both decomposition processes, but we also really thought that it was going to involve or be able to detect the signature of those above ground vascular plants. I mean, the plant response was so dramatic that we thought we'd be able to see a signature of lability in this system. And so we also looked at carbon quality through a measure called SUVA. And so SUVA is just simply absorbance at 254 nanometers, and then it's standardized by the amount of DOC in the sample. And when we do this, the temperature treatment completely disappears, actually. And DOC quality is being driven by lower versus saturated water tables. And in this case, higher SUVA values reflect recalcitrant material being lost from the system. So here we find under lower water table, we have higher recalcitrant material being lost from the system, and no evidence that those above ground vascular plants are contributing any labile carbon. So putting it all together, the dramatic and apparent changes in the above ground community, we really felt that they were going to cascade into the below ground community, particularly through root exudates, perhaps over the longer term through differences in litter quality and quantity, as well as these direct effects definitely on the below ground biodiversity, and metabolic constraints, reproductive constraints, allowing for increased biomass and activity, combined with the above ground changes, are altering decomposition rates, as well as just generally carbon and nutrient cycles. But, so whether or not northern peatlands are going to remain a carbon sink or a carbon store is still a bit of a question. But there's a subtler story here as well. Or I don't know, maybe it's actually the elephant in the room, and that's this repeated observation of our temperature treatments and the difference between elevated temperatures of +4 degrees above ambient conditions versus elevated temperatures of +8 degrees. And in all cases, what we found is that elevating temperatures by +4 were not significantly different from the ambient conditions. It was only after elevating all the way to +8. And so what this suggests is that there is actually a tipping point for northern peatlands under future climate change scenarios. We don't know where that tipping point is, if it's 4.5 degrees or whether it's 7.5 degrees or actually 8 degrees, but it definitely suggests that there's a tipping point here. Understanding when and how that threshold could actually be passed and the mechanism of it is sort of up for future work. And whether or not we can predict global change consequences based on this sort of alternative stable states model. So the changes in the above ground plants were dramatic and apparent. They were consistent with the predictions, at least in terms of the trends, but they happened quite rapidly. So, whether or not this is a realistic timescale, probably not. It's probably an artifact of the experimental design and being in a mesocosm. We thought that they would have cascading effects into the below ground. We definitely saw below ground patterns that mirrored the above ground patterns. However, again, the rapid response of both these communities suggests that there's similar drivers rather than direct feedbacks between these two. And lastly, that suggestion of a tipping point, we're definitely working towards elucidating where that threshold is and understanding the mechanisms that lead to perhaps a regime shift. And so with that, the next step is obviously to take this out to the field. We're initiating a large-scale experimental climate change study at 2 different sites in the same region. So you'll have to stay tuned for those results. Thank you.
Any questions from the audience?
Okay. In the first place, you showed really nice pictures of trees. So, my question is, how would the removal of trees affect your interpretation of changes in nitrogen and carbon cycling?
That's a very good question. And, obviously there's always limitations with mesocosm designs. We couldn't bring the trees back, mainly because I just couldn't carry them. We tried. But definitely you're right. And in fact, some of the results that are coming out of the fungal data, ASMA tells me that, of course, we are losing certain groups of her fungi. And it's not necessarily related to our experimental treatments, rather we're losing them because it's an artifact of the system and that we don't have the trees there to maintain the mycorrhizal relationships. So I guess the answer to your question is to be determined when we bring this back out into the field where we do still have trees.
Any more questions? Just one more.
Thanks for the presentation. That was quite interesting. I have 2 questions. Firstly, about you didn't mention about the elevated CO2 effects on the decomposition rate. I'm just curious if you see any effects of CO2 on the decomposition rate, because sheep fertilization could normally decrease the quality of leaches by increasing the C content in plant tissues. So I just wonder if you saw any effect.
Absolutely. We were actually very surprised at the lack of CO2 effects that we found. As I mentioned, we really only found subtle CO2 effects in a few of our plant species above ground. We found no effect of CO2 on any of our below ground measures. Now that said, we're hoping to follow up on that story a little bit more. One of the positive aspects of being able to do this in a mesocosm or in experimental greenhouses is that the CO2 that we use to enhance the CO2 actually has a signature to it. And so we're actually hoping to be able to follow that stable isotope signature and find out exactly what happened to that carbon. I think there are CO2 effects that occur below ground. I just don't think we saw them. I think they were swamped by temperature treatments.
The second question is about SUVA. You mentioned about the quality of the DOC, and I just didn't know about SUVA, so I was really interested. And if you could explain in what it is again, that would be great.
Sure. So I was new to SUVA as well. My collaborator is an eco-hydrologist, and so that's something that he brought to the team. So SUVA is a measure of carbon quality in terms of recalcitrance versus lability. And so it's absorbance of your DOC at 254 nanometers, and then it's standardized or normalized by the amount of DOC in the sample. All I can really tell you about it is that higher SUVA values infer that there's more recalcitrant material in that DOC versus lower SUVA values.
Thanks very much.
You're welcome.
There's one question there.
Your temperature treatment caused the system to change from a bryophyte-dominated system with no roots to a vascular plant system with roots. Are you able to disentangle from the results the effect of transpiration increases on the peatlands and that impact on water table?
So yes and no. So our water table treatments were very strictly maintained, and so in this particular experiment, we wouldn't have seen them. The mesocosms were being watered twice a week with a dilute nutrient solution, sort of thing. We do have some follow-up data that we haven't analyzed yet, looking at NEE and MPP and things like that. Sorry.
Very nice study. I do have a general question about the experimental approach. Global change would occur over time slowly. So what's the role of adaptation compared to the way we're doing experiments? 4 degrees to 8 degrees is going to take many, many years. Thank you.
Absolutely. And I think, that's a well-recognized a factor here in how we're approaching global change experiments. Of course, it always has to be paralleled with logistics. We can't have an experiment over 100 years to see how the system's actually going to respond. So, is it reality? Yes, no, maybe. The actual values that we're getting out of it, I don't think are necessarily going to be absolutely bang on. I wouldn't want to put together a carbon model based on this. However, I do believe that the trends, the direction of trends, and the relative effects of the different treatments are still real. So even though we can't create a realistic scenario of global environmental change, I still think that these experiments are valid and valuable.
Similar to the last question, one of the things that you showed was a change in a reduction in variation with increase in temperature, and you mentioned that it was driven by a greater relative abundance of few members in the fungal community and the microarthropod community, and I'm wondering if the organisms that increased in relative abundance are organisms that tend to respond better to stress.
Respond better to stress? Perhaps not. If anything, they were able to respond to the actual temperature treatment. So as I mentioned, they, at least in the microarthropods, they were small-bodied, asexually reproducing, and this also coincided with a large increase in the proportion of juveniles. So I'm thinking it's more metabolic and reproductive constraints that are sort of released off some groups, but perhaps not the larger-bodied sexual species.
But so it could potentially be the speed of change could trigger the particular increase in relative abundance of some of the more dominant organisms. Absolutely, yes, I would agree. Yeah.
Just one quick question about your tipping point.
Yes.
That is very, very interesting. I was wondering, it's because when you saw your pictures with the different stages of your mesocosms, and you said increases vascular plants. On the last one, could I see a shrub in there, like a heather or something like that? Because our recent work shows that shrubs in particular make a different change. So I wonder if your tipping point is when the shrubs appear on your systems.
We're attributing the tipping point to the graminoids. Originally, we were actually expecting it to the elevated temperature and CO2 effects to be on the shrubs. So we had a small Chamaedaphne shrub in each of our mesocosms. But I think the time scale that our results occurred at were really about the graminoids. I think that the shrub effects would still be there, but they were emerging over a much longer period of time, and the graminoids just sort of swamped them out.